Hello, everyone. I'm Qitian, a postdoc fellow at Eric and Wendy Schmidt Center. And I'm excited to introduce our speaker, Eric Xing. Eric is the president of Mohammed bin Zayed University of Artificial Intelligence and co-founder and chief scientific officer of GenBio AI. Prior to this position, he was a professor of computer science at CMU and the founder of Patton. His research focused on the development of machine learning and statistical methodology and large-scale computation systems and architectures for solving problems involving automated learning, reasoning, and decision-making in high-dimensional, multimodal, and dynamic possible worlds in artificial, biological, and social systems. He is a board member of the International Machine Learning Society and fellows of the IEEE, IEEE, and ASA. Eric, I'm excited for your talk, and the floor is all yours.
Well, thank you very much. I want to thank Caroline and the colleagues for inviting me here. I had really a feast of, learning, for the past 2 days, learning all these very wonderful, exciting projects and ideas, which actually pushed me to edit my slides almost constantly because I feel like the stuff I prepared to talk about can be easily outdated by the new presentations in here. So I try to make it more fun. In fact, just a minute ago, I added a few more slides just to give maybe a fun context of this. So we are here to solve very complex problems. In a sense, it's a form of complex reasoning. And I heard the word LLM has been mentioned all the time. And LLM has now been also a backbone for many of the applications that is mentioned in here. In fact, the architecture was directly borrowed to train DNA models and RNA models. But I think there is a need to really take maybe a step back and to look at what is this whole business is about. What is LLM and what's the potential that we might have ignored? So LLM is basically a system that does reasoning inside the space of language. It is actually not meant to be built to make chatbots or to make linguistic functions. Okay, it is actually originally built to be a next word generator that is meant to simulate linguistic behaviors. Basically, it is a success when you feel what you get out of an LLM interface does not look different from a human. So it is simulating a human in the space of language. And the LLM doesn't work when you add a space to make it bigger, to have images. And when you have adversaries and other agents, you add another degree of dimensions. And we are here. In fact, we are now in the space of study nature laws, right? So the problem becomes really, really complex. How we might approach it? What is the kind of fundamental spirit inside large language models and foundation models that we could even borrow, but not necessarily just directly use to attract and to attack bigger problems? So I hope some of you who are like myself, a sci-fi fan, have heard about the story of Dawn. Have you heard about that? Right. Dawn is a super intelligent agent which solves larger problems, like all of us, larger problems. They solve problems like winning a galactic war, transforming the climate, and turn desert into greenlands and so forth. So these are problems really difficult, right? And one thing very interesting in the book is that he solves the problem in a way that is very different from we physicists and mathematicians who are doing linear thinking. We have the inductive reasoning machine to drive through very, very logical inference. He's doing simulation. Basically, he would have a mental world that allow him to simulate all possible plans and ideas and approaches before he even take a single step. And then he would be able to choose the best scenario and then action on it. And the reason he has that capability is because he inherited all the experiences and knowledges, and meaning data, from his ancestor. And the more he inherits, the more simulation he has, and then the more power he can use to solve problems. Right? So in the world of psychology and philosophy, there is actually a term for such way of thinking called the hypothetical thinking. We actually do it all the time. We say we do thought experiment. That's exactly hypothetical thinking, right? I'm not sure that in our daily research, In fact, among the talks that we heard about, we do thought experiments that much. But what I'm going to talk about today is to basically build a system that hopefully allow us to do simulational thinking and thought experiment in the world of biology. So in a sense, the project is about building such a function, which could be called a word model, if you are in a different study domain. But in here, I call it a virtual organism. It is a distribution of the next state, given the previous state, conditioning on also action you take, which could be perturbation, for example, knockouts and other things. And that's here I want to conclude the primer. The models released like this. They are basically the same thing, trying to simulate phenomena and the possibilities in different space of the world. LM, lives in the space of language. Word models lives in the space of space and time, and agent models lives in the space of a multi-agency, and the ADO model that I'm going to talk about lives in that space of a physical, biological world. So I'm not trying to solve a model which make a good prediction of perturbation outcome or make a good prediction of a structure given the sequence. These are byproducts. The real goal is to be able to simulate biology, the way that you would expect, from a natural creature. But the goal of explaining why you simulate that way, what's the mechanism behind biology, is actually not in the goal. Okay, it is not the purpose of such a project. So with that, I want to get started toward an AI-driven digital organism using a multi-scale foundation model approach. So we've heard enough about this in the past few days. The living creature is a very, very complex system, highlighting multi-scalability and multimodality, and it's a holistic system. So I'm not going to say anything more than that. You need to be able to understand and process information across the scale. And that's actually not traditionally, even until now, how biological science and medical science are actually positioned. For example, how do we study biology? If you are given a piece of tissue which has a cell, in our school, we have the Department of Cell Biology. And the scientists would focus on that, take data in that domain and study that. And there is also a department of molecular biology who goes down, which I used to get my PhD from. And there is also biochemistry, which go even one level down, right? If you want to go up, you have medicine. If you go to a hospital, you see divisions organized by organs, okay? If you have a liver problem, you are going to that division. And therefore, they're not looking at your other organs and other tissues and other things. And, we also have public health and other things. But we use the knowledge in those research to solve, say, this kind of problems, drug design and target discovery, coming up with treatment and so forth. So maybe you can already ask yourself, does the solution to each of these problems exist in one of the slides or not? Right? Maybe not. Right? You probably are expecting something like this. Each of the problems cross basically that sector or that silo that defines our research discipline where we get the degree from, which kind of forces us to look beyond where we are. But our ability to look beyond is limited by tools and also by our brain power. We may not be able to store so many knowledges. And that's where LLM all of a sudden kick in, because they seem to be a good tool, at least to begin with digesting and consuming all knowledges, right? And it is maybe also echoing that very reality that every human healthcare problem is essentially a biological problem. If you really talk about someone being sick, you want to come up with a design of the drug and antibody, it's not just a medical problem anymore. You need to know the biology, at all scale, anywhere from molecular behavior to cellular behavior to tissue to organ to organism. Right? So, if you really want to see your answer in that holistic context, I do see, the need to rethink about the tool we use, which is like this. Our classical mathematical tools, which have been with us, for decades, and we spend career and life, developing higher and higher version of that, is actually of that nature. It's under the principle of one model for one problem with one data set, that kind of specific design. In fact, we are very proud of advocating that spirit, that you need to be very specific, okay? And you shouldn't be trying to be a generalist, because that's going to cause you losing performance and so forth. But in terms of correctness, that's the second issue. In the recent years, we see a new generation of technology, maybe featured with deep learning approaches in the form of convolutional neural networks, transformers, and now bigger foundation models. How are they going to be, maybe used, maybe in a different way to address the problems that biologists and the medical practitioners might be interested? Well, we see some very good success already, right? So AlphaFold is one of the highlights of using these new tools to solve a very, very classic and difficult problem in a very, very impressive fashion. And as a result of that, these days, we see a boom of foundation models. In fact, it's even more active than the space of large language models and word models. Almost every week, we see announcement from certain labs of a new model in a particular modality and so forth. Right? In fact, you may wonder why so quick. Why you only see a foundation model in language and in visual only from big companies and only after a couple of weeks or maybe a year, and why we can see biological foundation models coming up every week. In a sense, my humble opinion is that these are not really foundation models. Okay, these are transformer models, which are smallish, specialized. It's just a bigger classical tool happen to be adopting the same architecture. You don't call a bicycle, a real kind of same beast as a 18-wheeler, even though they all have the wheels. It's a small thing. And also the design philosophy probably is also very different. For example, imagine that we use a tool that we are doing now to solve a simple, say, structure prediction problem, right? You probably can see that the structure prediction problem right now is happening out of the biological context. You take a amino acid sequence, you put them into let me say AF3 or other foundational tools, and you get a structure out of it. Here I give a result for you. This is a comparison of the ground truth of a slightly complex structure prediction problem, which you need to consider context. You are predicting the structure of an antibody in the presence of an antigen. Therefore, it's not just about your amino acid sequence determining the three-dimensional structure of a protein, but also it needs to work on the antigen, which actually will induce additional structure consequence and so forth. It turns out that AlphaFold 3 didn't do very well for this sort of task. Okay, this is called a complex prediction problem. In fact, the problem can be even trickier because not only your structure needs to interact properly with the antigen, it also shouldn't interact with something they're not supposed to interact with. Therefore, the off-target problems, the non-specific prediction interaction problems should also be in context, so that you don't trigger toxicities and other side effects when you deploy a molecular design. So in a sense, what I'm trying to say is that even for a biological problem, which look as specific as a from sequence to structure, you need to consider a much bigger context. Therefore, that so-called one model per one data set, per one problem kind of mindset isn't going to work very well, okay, for a complex problem like this. Right? So that's why when you look at large language models, it was never trained in this way. It is not like I train the large language model to answer your Olympic math problem. I train another model to answer your reason problem. It begins the reverse way. I train a generic general purpose model, which is humongous, very, very big. And they don't perform very well, but they actually does work for everything. And then only when you want to do your math problem, you want to do a financial prediction problem, I specialize it and fine tune it. That's the workflow in LLM. But in biology, we are not doing that. We actually are treating the foundation model as an end tool already for a specific problem. And I feel a need to remind everyone that this may be problematic. We are missing opportunities. So in that sense, I would say not every foundation model or state space models or combinations is really truly foundational, okay? And the LLM is never meant to be doing that kind of a task-specific fashion. So then what should we do? So that leads to my very humble proposal where I want to welcome some debates and inputs. So we've got all this data, right? This data include multimodality data from a sequence to structure to tabular record of expression and to image to time series and everything, right? And we've got all these questions out there, all the way from designing a protein structure, reverse folding, a protein sequence, and all the way up to, I don't know, deploy a antigen or a vaccine, at the proper time, at a proper dosage to a proper population and so forth. All these problems look very different, but they have a common foundation of following some biological principles, and also they may be, reaching out to solutions that is at this level connected, right? So in that sense, what should be in the middle? Should we see a jungle of spikes which connects one thing here to something over there? Or do we maybe imagine maybe a future solution that is more modern down the road, is taking a more holistic approach, right? So I believe that there may be an opportunity to think about a more unified and integrative system for foundation models biology, where they can do one thing right to simulate biological systems. And through that simulation, hopefully you can make predictions, you can make programmings and designs, right? So that's a system where I call a digital organism. It is basically a computing system which would empirically, simulate the phenomenon and outcome of a living system upon any of your, legitimate and implementable perturbations and interferences, as if you are interacting with a real world organisms. So I have some very, very strict definition here. It's not a hypothetical thing, say, you do everything for me. First of all, you need to be able to ask and execute your ask on that system. Don't basically try something that is spiritual and say, tell me how do you work? Because this is something that you cannot really execute and implement, right? So typically you'll have a prompt, which could be a sequence, could be a structure, could be a knockout, could be some kind of perturbation, and then the system hopefully simulates you back, whatever consequences. So that's a digital organism in my definition, which is not a digital twin. They don't necessarily look like a cell, but the numbers that they give you may be what you would expect from measuring a real cell. And in the essence, this system is essentially mathematically equivalent to such a conditional distribution of the next state, given the previous state, and also the action which represents your perturbation, right? So the host just introduced one of my functions to be the chief scientist of GenBio. GenBio is a company that we actually recently created exactly to execute that vision, because it requires not only very, very intense engineering work, but also resources and machines and so forth. So GenBio's vision of a AI-driven digital organism is illustrated in here. It is a holistic family of foundation models, each built on a different biological data modality, but they will work together at some point to deliver a holistic, multidimensional and multifaceted functions. And we are hoping that such a system can be used to augment not just a single slice, but a spectrum of every stage of processes in the biomedical medicine. For example, nowadays, I remember very recently, FDA just announced that there is time, maybe it is time to start now stepping away from using animals and real systems to do all the trials. How to make that happen? Well, that means you need to do something in a digital environment with simulations. So that means the AI-driven innovation in medicine is not just about simulating or designing a drug structure. you need to also simulate the environment where drug will be acted on and hopefully predict their consequences. And you may also be able to help designing the trial, given such context, make it more efficient and more informative, and maybe even helping personalizing the outcomes. So we see such a system called ADO, potentially usable for needs of across the spectrum. And our dream is to really reach to this point, to close the loop of biological study. In the past, I was actually trained as a biologist. I remember that in my whole 5 years of PhD study, I studied 3 genes. I was taking pride of being able to pour a beautiful gel, and get a sequence out of it. And then we use that method to basically do biological exploration of the hypothesis. We basically make hypotheses, and we make some constructs, and then we are going to see in real life, how that perturbed system will look like. This is a very expensive and dangerous kind of experiments because it's in the physical world. What if you have a leak, you have a spill, right? So I imagine with an AI system, the whole workflow will be turned upside down. We are going to probably, spend instead of now, using data and computer as an afterthought, but using them as the primary filter, to search the hypothesis until you, reach to a few very highly plausible hypothesis, using computational simulation, and then you do a much more guided and small-scale wet lab experiment, and then hopefully use that outcome to improve data. So that's basically what ADO vision at a very high level look like. Now for the rest of the talk, I'm going to dive into the technical details and tell you how such a system might be built. It's a very complex system. And for every complex system, if you take an industrial view, you have to basically come up with an engineering roadmap. And our engineering roadmap is no different from building a big aircraft. Just take into parts and do distributed manufacturing and then assemble and fine-tune. So stage 1, very obviously, it is where we are now. We divide and conquer. Basically, all these existing efforts that we see in the literature, in a sense, belongs to this stage. You are building modality-specific foundation models, for DNA, RNA, protein, RNA-seq, image, and so forth. Yeah. But why we need to build that rather than just collect them from literature? Because we want to see all these different components, first of all, to be performant, but also to be ready to integrate, because they need to talk to each other so that a problem can be addressed in a bigger context and also information will flow. Therefore, our next step is to really connect dots. And this is the technically more interesting part because different models use different data and they have different representations of different modality and maybe even of the same modality. That creates the need to seek alignments across different modality to also evolve the model continuously when you get more information, and also to maybe allow arbitrary conditioning to be implemented so that you can do forward generation, reverse generation, or coming up with correct responses to your creative prompts. Say a particular drug situation, structure requirements, temperatures, and so forth, and then cascade. Maybe I should replace this word with a little bit technical description so that the people who are here working on the models can maybe appreciate the substance. What is evolving? For example, you have a language model that's trained on unaligned collection of sequences. Now, what if I give you additional information that your sequence can be aligned into multiple sequence alignment? Do you retrain the model from scratch, or do you continue? And how do you continue? Right. So that's another interesting problem. And also maybe you get more data of a completely different distribution that you need to maybe take on a non-trivial way of weighting them so that they don't spoil the previous trained models. So this is evolving. You need to basically allow the model to continuously receive new information that is cleverly engineered and formatted to allow the model, keep learning without disrupting in the overall architecture. I will give you a few examples when I come to that point. Second is about the alignment. Yeah, your protein sequences come from amino acid sequences, but also could come from the structure tokenizations of structure discrete motifs. And can you actually cross predict each other so that your representation become aligned and consistent? So this is an art of alignment. It's like you put two languages next to each other. Therefore, your representation of the language in the language model becomes suddenly connected. Conditioning, I don't need to say too much. If you do inverse folding, you input a structure, then your production of the sequence need to be conditioned as structure so that they become consistent. So our goal, of course, is to allow arbitrary information to be encoded properly so that they can be conditioned on. This is particularly important when you do cell perturbation. How do you embed and represent a perturbation, or maybe a knockout, or genetic perturbation versus drug perturbation? Right? All these could lead to a new need of architectural innovations. Cascading is another interesting information. That actually may be one of the best justification of having all these different models in the same place. Why? For example, when you build a cell model, how these days people build a cell model? Well, you use a table of RNA-seq data, and therefore you train a model using such tabular information as the input. And how do you represent your gene? Well, a gene in those models is an ID. The ID could be A, B, C, could be 1, 2, 3. It's a code number that is not carrying biological meanings. But a gene has DNA sequence. A gene encodes a protein which has structures. How do you put all those information into the learning so that they carry both just a information that tell them apart, but in addition their actual functionality? Well, you can imagine once you have a DNA foundation model, once you have a protein foundation model, you can extract representations of every gene and every protein from those models and use them to represent the genes, the protein, the genes in the cell model. And that cell model itself actually could return you a representation of a whole cell, which can be used in the next level of modeling, say a tissue, or organism. Right? That's how you see the cascading can happen continuously, top down and bottom up. Once you have that, we can write at the age where you need to do optimal alignment and across the scale. It's just like when you assemble all the parts into an aircraft, you still need to do a lot of fine-tuning to make them harmonic and working together. So in here, that poses another technical challenge, because that means you need to be able to co-train now multiple models at the same time so that the gradient can flow across modality and across model modules. And also there are additional consistencies to be honored in this fine-tuning. So that's roughly the 3 stages that we are using right now to train the ADO model. There are, again, a good number of technical challenges that we are working on, both front end and the back end. I just want to name a few. On the back end, these models are bigger models. I'm going to have a graph with you showing you that why we are not at the optimal spot yet, even in terms of model size. Therefore, that involves handling long context, a million or maybe longer, and running on tens of hundreds, if not thousands of GPUs, and doing proper parallelizations, doing proper discretization, quantization, and other type of projection. So these are the level of engineering work on the back end. Then on the model part, the need to invent new representations for biological information, which is the business called tokenization, is not end yet. Right now, in fact, we don't have a lot of new ideas coming straight out of biology in treating biological data. We basically borrow all the ideas from NLP, which was meant for cognitive languages, not for biological languages. Therefore, you can imagine there is a big room there to improve our current way of representing even biological information in the form of tokens. And then positional encoding, what does it mean by position in a biological token? In the language, it's very obvious. It's the position of your word inside a sequence. But in biology, that kind of assumption is not there. Therefore, right now, we see many trivial treatment of positional encoding, which actually lose huge opportunities in capturing additional informations. And also, training strategies. Do you still want to do the missing token prediction training, or you use contrastive training, and so forth? Again, that gives you additional dimension to make the model perform better. Inference, how do you use the model after it's trained? And I think especially in those generation tasks where you need to have a particular structure serving particular goal, in the space of doing agentic and the word models, now one of the trendy terms is called the purposeful reasoning and the purposeful planning. So that's another idea that is not very often practiced right now in biology. What's the purpose of a design in biological world? How to encode our purposes? So I meant to say that it's a very, very exciting field that is almost empty at this point in terms of technical new ideas. So both for machine learning people and also for biologists, it's a gold mine for us to discover new technologies and new advancement. And now, of course, there is a huge need there for self-engineering. In the space of LM, we talk about the Megatron database, we talk about the deep speed, all these other things. In here, the tooling of LM of biological modeling is really, really chaotic right now. I saw a few presentations today and yesterday mentioning about certain companies already making that effort. It's nice to see more of that coming out of our work. So just to report some preliminary results before I end, we were able to publish a good collection of pre-trained models already in the past few months. And if you go back to this graph, all these little components with a check mark already has a model released, and some released more than once already. And of course, they are evaluated very exhaustively over hundreds of different downstream tasks to make sure that they are performant and also consistent. What do we do with all these models? What have been happening in developing this? So here are some technical insights. First of all, I want to report that we did scaling law for many of these models. And we found that even with the current data, the model size isn't saturating yet. You still have the room to push up the performance of the model just by making the model bigger. That's a kind of very, very low-hanging fruit that we can try. Of course, it requires capital and engineering capabilities. There are some more interesting technical advancements, which I'm going to quickly list out. For example, in this ADO protein model, we start to see the opportunity of very, very deploying diverse pre-training strategies to constantly, continuously boost the model performance. So in this particular production of the protein language models, we started from single sequences, and then you add MSA. The MSA actually can be treated as a single sequence if you inject a positional encoding which indicates the evolutionary position of every sequence, and then put them back. They remember not only who you are, but also where you are from the evolutionary history. And that essentially turn a previously single sequence-based LLM into a model which based on MSA. You can then inject stratoconization to further improve. You can then play with data reweighting, temperature annealing, and many other tricks to constantly push up. So you can see this curve is still not stopping yet, which actually gets us to a point where we can outperform models like AF3, ESM quite significantly over protein function predictions. In another model, which we call the ADO-fold, again, a combination of different ideas really can lead to quite strong results. I mentioned about the prediction problem out of context. And here's how we did it. First of all, we rely on a much, much more powerful protein language model that features ideas like multiple sequence alignment through pre-training, a mixture of experts to allow you addressing different model modality, RAG, to really allow you to focus on, connected data, and then, some diffusion process, to allow you, simulating structures. And we found that with this combination, we were able to actually predict, a complex antigen-antibody structure, in a much, much higher quality than existing models. In particular, if you look at these three graphs, this is the prediction quality comparison between us and AlphaFold and another model, over only antibody, which is basically one complex structure prediction problem. And then in here, the task is performed on antigen and antibody complex. And you can see the improvement is now over 20%. And also nanogen and nanobody and antibody structure prediction also is a hard one. We can do much better than existing models. Basically, the harder the problem, the better improvement you can get from such a slightly more complex systems. Yet another innovation related to the diffusion-based generative models. Here, again, you can see that we implemented a pipeline which allow you to do reverse folding, of the protein structure, given a target that says generate something that binds this particular structure. We found that our system was able to generate a much more realistic distribution, of the protein structure. So here is a comparison. This red curve is the distribution of native proteins in terms of the proportion of alpha helix and beta sheets. This red curve is the distribution of the generated products from David Baker's group. And here you have another function. You can see that they are very far away from this red thing, right? And this blue curve is the generative distribution from the ADO diff gem, which appear to be a lot more faithful to the biological reality. One more is on the ADO tissue model. We built a cell and a tissue model which can be tested on problems like niche prediction. But the way to do that is more than just building a cell model, with single cell RNA-seq data. We actually use another new technique called the two-dimensional positional encoding, so that whenever you want to build a representation into the prediction, you take not only the cell itself, you also take the cell embedding from a neighboring thing. Basically your neighborhoods, which are connected, to you, will all be incorporated into a much larger representation, so that you have the context, into that. Therefore, in training this model, every count is not just from one cell, but also the cell in the neighborhood properly positionally encoded. Last but not least, we actually could use this entire idea of foundation models to redefine some classical practice, such as GWAS. Right? So in GWAS, it's a very, very important field in medicine, but it's also really, hampered by technical difficulties. You cannot, for example, work with very fancy phenotypes, such as a time series. You also rely on very, very statistically weak data in the form of SNPs. Right? But now, with a foundation model that works across modality, first of all, you can turn your complex phenotype, such as an image, such as a time series, into a vector representation using the foundation model embedding. You can also turn the genomic information into a continuous vector, which can separate all the polymorphisms and the behaviors and patterns in your genetics in the form of a vector. And then at the end of the day, what you do is vector-to-vector association analysis, which is a multidimensional regression problem using classical approaches. So that kind of opens up the way of arbitrary flexibility of working with different type of phenotype modality and also genotypic and molecular polymorphisms. And all the results I just talked about are actually packaged in a nicely structured software stack where we regularly release in Hugging Face and in GitHub. And you are welcome to try out and to give us feedbacks. I think recently we've been keep an eye on the downloads. It seems to be picking up quite nicely with thousands of downloads in certain models. So I definitely encourage you to take a look. So to wrap up, I think I'm maybe sending some slightly controversial messages in here in terms of my methodology belief, right? Because we were trained as scientists, and we believe in ration and the math and the first principle was supposed to be like Newton, writing the laws and predicting the orbits and so forth. That's actually where scientists really hold very sacred about. But I want to say that this may not be the only way to do science, because even in science there are many phenomena which is defining causality in a traditional way. Here I just give you a few examples. If you are a believer of a general theory of relativity, then there is a phenomenon called time travel. Therefore, what is causing what is unclear there. Okay, quantum superposition is another problem. Only when you observe, the final consequences. Otherwise, there are parallel universe out there. Therefore, there is no causality, right? And the evolution also is not causal. By the way, we are not the perfect design of the God. Okay, we are the outcome of a random mutation plus a directional selection. Therefore, even in nature, it's a hypothetical thinking type of a process rather than a direct design. I want to design a creature which lives in the mountain of Alps. That's not the way we evolve, right? So I just want to maybe stress that there is a room for us to be a little bit less rational and more empirical, and that basically comes to the conclusion. Between the two camps of school, right now, Kantism, is maybe the kind of a guiding principle, for much of the scientists. They always ask why, and can you explain to me this? And I need to have a pure, total clarity and causality before I can believe, and move on. But in humanism, you can take your face on data and the empirical results before you even understand what's going on there. That's actually what's inside LLM and large language models and FMs. We actually honestly don't know exactly the reasoning path and everything inside that. But it doesn't hurt us from using that. At least in biology, I find one example similar. The invention of the vaccine is like that. When it is invented, we don't actually know DNA, RNA. We don't know at all why it is working, but it worked. And then people used it, right? So I hope that we can take the same kind of approach in here with the availability of foundation models, which does a job like this instead of this. I would like to have in a position where we have a periodic table and predict the compound, when we mix them. But I guess even if the FM-based approach, the deep learning-based approach, the AI approach, are not quite there, it doesn't, prevent us from using it to do, to make progress. So with that, I want to return to the initial vision. ADO is one of these simulator, of the biological world, allowing all these phenomena to be simulated and some of them to be programmed and predicted. And there is a potential that such a system could be, a paradigm for holistic and in silico science, where you carry out your virtual wet lab, you carry out your virtual trial, and you do, maybe pandemic simulation using a simulation system like that. Like what we actually see in many other domains, in physics, in semiconductor, in agriculture, in many other places. It is happening, or it happened for a long time. Biology is one of the last places where we are very, very heavily still, relying on wet lab. And I imagine a change toward a modern approach is needed. Okay, with that, I want to conclude my presentation. Thank you for the attention.
Thanks for your inspiring talk, Eric. And we'll have time for one question.
Hi, thank you for the talk. I have a question. Have you faced any challenges in terms of evaluation benchmarks? I would assume this is like a novel idea you're exploring, and I would assume you have faced some challenges. Are you working on creating new benchmarks for this task? Thank you.
Great question. So fortunately, just like in ImageNet, in the biological world, we have, for example, protein gene from Deborah Marks, which gives you already hundreds of downstream benchmark tests. So we use that. Okay, and I think there is always a need for the community to share and publish more standardized benchmark tests. But there is a twist going forward. These benchmarks were designed with the old mindset of having new tools delivering old functionality. They may be not strong enough to test the future systems. Therefore, I think with the evolution of new systems like this coming out, there may be a space to think even harder to come up with additional, stronger tests and more difficult problems. So that's, of course, our company, my team is very committed, to publish and to share not just the benchmark data and tests, actually the benchmark tool. You need to have the right tool to plug in your data and run the experiment. Even that right now is not very standardized. This is not like you have a chatbot arena, in LLM, which actually was built by my group. Everybody go there using the same platform, to fairly compare. I think this is an emerging need that we need to address in our biological community. Thanks.
Building a parallel model, just very nice presentation, and you have wonderful, very platform. But not all health issues are biological. It could be biopsychosocial in 3 dimensions. For instance, like addiction, can only account the genetic only account for 40 to 60%. And practical guidance, environmental, epigenetic, all come into the equation. Like whether in the future would like to entertain a parallel model or additional model on top of your biology.
Thank you for the question. If you look at this graph, this is not meant to be setting already the limitation of the data we're going to use. These are examples of the data we have. And if more data is going to be available, such as epigenetic, such as environmental, of course, we're going to use it. The key question is that, do you already design an architecture that is maybe ready in advance for taking those big data, new data, or do you just plug one after another, right? So this is a technical question. We don't have an answer yet. But if you look at the practice in large language models and in large visual models these days, the attitude over there on data is very different from biologists. I keep hearing in biological domain, people say, we need to have a high quality data, we need to curate the data, and we need to do something. I have a drastically opposite opinion, maybe just to be sarcastic. All data is data. Wrong data is data. Dirty data is data. They carry informations, and if they reflect a flaw in the experiments, in the collection, that is something you want to learn as well. Therefore, in language, for example, they just take everything, okay, and let the machine, the system, to try come up with their own internal connectivity and coherence, and then give you the simulation. I think in biology, if you take an open view like this to try make the model larger and make the model inclusive of all data, that might be an opportunity to see certain emergent properties.
We'll unfortunately have to move it on to the discussion.
Thank you very much.
Thank you very
much.
much.
Sorry for taking your time.
Thank you.
Thank you.